Can you hear me? Okay, so thank you, Sarah and Madeleine and Iker as well for inviting me to give this talk. I will try to explain you how important sponges are for ecological reasons, but also as tools, because we use them to monitor marine benthic communities. Let's see. I don't think I have to remind you that the effects or the consequences of climate change that we have currently that we are currently experiencing in the ocean. Examples are just ocean warming that has consequences in coral bleaching disease and massive deaths of numerous invertebrates and also vertebrates as well, shifts in distribution ranges and shifts in community structures. This is just some of the effects of climate change that we can name in the ocean. And all these are obviously causing biodiversity losses. But the truth is we don't know, we don't have timelines or good estimates of the biodiversity with a timeline for many species. We do for many vertebrates because that's the general bias in the scientific world, I guess. But for invertebrates, we have just very good estimates for, or relatively good estimates for coral reefs or for corals. But the rest of the invertebrates are a little bit underexplored or forgotten sometimes. But what we know from these estimates is that 58% of the coral reefs are potentially threatened right now by human activity. And we know that climate change is causing biodiversity loss everywhere. And as I told you, we can do, we are able to protect what we know. And so most of the work in protection and conservation has been in the past years focused on coral reefs, but through these marine protected areas that are the most effective tool that we have for protecting ecosystems. The problem is that coral reefs are not the only ecosystem in the world, even though they are extremely important. There are many other ecosystems that we don't have all the tools to protect because we don't know them as well. And one of these places, ecosystems or areas in the world are the high seas. And one of the reasons that many of the areas that are very important for biodiversity reasons in the high seas are not protected is because nobody knew where the responsibility lied in these areas. But since a couple of years ago, we have a treaty that says that regulates the activities, the fishing, the mining, and assign responsibilities for the different nations that have interests or have a connection to the high seas. And the goal is to actually protect 30% of the world's ocean under the protection by 2030. And I might be a little pessimistic or not overly optimistic, but I don't think we're going to make that goal because right now only 1% of the high seas has been under any protection. Basically, because to protect something, we know we necessarily need to know what is living in there, right? Because not only for in terms of biodiversity, but what are the connections between the different populations in the different ecosystems, how these ecosystems are functioning, and what are the levels of genetic diversity in terms of understanding the resilience of these the organisms, not animals, everything, right? So one of the hurdles for this is monitoring, biodiversity monitoring, because it's obvious that in places like these beautiful and warm seas, like in the Caribbean or in one where Sarah was before, it's relatively easy relatively easy to monitor the biodiversity through transects, photos, or whatever. But there are more remote areas that are not so easy to monitor through traditional surveys. And one of the most important limitations that we have right now is taxonomic expertise, because I work in a museum and I know that taxonomy is almost dead, right? It's not, I mean, we're trying very hard to not let it die, but it's very, 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 very hard. And one of the reasons is because the work of taxonomists is not as cited or recognized as the rest or many other scientific. So one of the things that I usually say in these seminars, in these talks, is that we need to cite the taxonomic work of the species that we use in our papers, because otherwise they will not get a portfolio, a research portfolio, as productive or as relevant as other scientists. So it is true that the traditional surveys need a lot of taxonomic expertise to understand what we have in hands. And sometimes in the ocean, not sometimes, but many times in the ocean, many species are not even described. So it's really, really difficult. It's a decade-long job, I would say, in many cases. And there are some scientists that decided that science or biodiversity monitoring cannot really wait for that long if we need to protect some areas. So that's why one of the tools or the mechanisms for assessing biodiversity they realized they could use is through the study of DNA. And in this case, through the use of metabarcoding. And they designed these autonomous reef monitoring systems that are basically some plates. These are replicable across the areas or across the different oceans that you want to study. And then you use some of the plates to understand what has settled or what is living in the plates, and some of the plates you scrape them off and you put everything together, and then you do a DNA extraction of all the material from the different organisms, and then you amplify different genes that are markers for the different species inhabiting these plates. Then you do the regular pipeline for metabarcoding, for sequencing and processing, and then the analysis of community composition. However, even though they are extremely useful for some organisms, they have tremendous biases. Two species that can actually live in these relatively small plates, or that are more prone to establish themselves rapidly, whereas some others are less quick, I would say. So there are some biases here. So that's why some other scientists decided that maybe we can recover the DNA that it's in the water that has been released through metabolic waste or free DNA coming out of the cells, skin, scales, excretes of the different animals, or even tissue coming from wounds, etc. And they decided to just filter water and do exactly the same thing using different pipelines. You can do qPCR for quantitative assessments of different organisms that you have in the water column or in a certain ecosystem, or you can do metabarcoding through next generation sequencing using different primers to understand different types of organisms, because not all the primers have the same affinities for the different organisms. Obviously, if you are interested in looking at the fish communities, you would have to go for my fish primers that target the 12S. If you're interested in the whole community, obviously you need to go for CO1 primers for cytochrome oxidase C1 amplicons, but they don't work equally well across organisms, and the same for 18S. But if you're interested in the microbiome, there are some protocols very well established for microbes using the 16S, and I would say these are the most robust primers of all. But obviously, the eDNA coming from the water also has some biases, and their biases are related to the affinities of the primers, but also they are related to the actual volume of organisms that you get from the water when you filter. You're going to get a lot of different algae, a lot of different planktonic organisms, microscopic planktonic organisms. So your estimates are going to be biased through to these organisms, especially in surface waters, where most of these studies have been done right now. But the truth is that the estimates that eDNA can provide about the richness of a species in a given ecosystems are moderately, let's say moderately, similar to what are obtained through traditional methodology. So it's a robust method that needs some tweaking, of course, and some more development, but it's going in the right direction. And it opens possibilities as well for doing some other things besides biodiversity monitoring, like doing metaphylogeography, because you can actually recover different haplotypes from the same organism across areas. So that's a fantastic approach that you can take using eDNA approaches. And also you can do fish stock assessment. This is in the infancy. Obviously, you can do this through qPCR very accurately, but the estimates that you get from eDNA in terms of number of reads assigned to a fish species are not so perfect. Sometimes you get a lot of reads for very small fishes, and it depends on how big the school is or whatever. And in the future, and this is actually coming now, we can even reconstruct trophic networks for the different ecosystems. Sorry. But as I was telling you, there are some taxonomic biases. These methodologies are hard to standardize because you probably are aware that it's not the same filtering 2 liters of water in a pristine coral reef than doing 2 liters of water in a reservoir that is basically it has a lot of sediment in the water, so or lots of organisms that are going to saturate very quickly your filters, right? So it's very hard to standardize how many water you get, what are the filters that you get, and all these things, what are the markers, etc. And also there's another thing, that another limitation is that DNA degrades rapidly in the seawater column. It depends on the temperature as well and the pH and everything, but it degrades relatively rapidly. So these eDNA methodologies through the water, using water, are relatively good or very good for planktonic organisms or organisms living in the water column. But what about the ones that live in the benthic areas? The estimates from eDNA from the water are not so perfect because some of the organisms, the sessile, for instance, sessile organisms that live in rocky benthic areas are not releasing a lot of stuff or are not releasing as much DNA to the environment as some other, like fishes or things like that. So some very clever researchers decided or tried using natural samplers. So for instance, using mollusks or mussels that naturally filter the water, and obviously there will be a lot of environmental DNA in this water, and these guys accumulate this material. So they try to use them with relatively good results. But there's some biases as well. And for the sedimentary animals, they try to use shrimps. But again, there are some species you can see in these Venn diagrams. There are lots of the species or several species that are shared between what you can recover from the stomach and from the actual sediment, from the stomach of the shrimp and the actual sediment. But still, there are some that are not recovered. And these are mostly focused on recovering fish communities, which are very important for the ecosystems. But who is the best, I would say, filter in the ocean? Obviously, the sponges. So we decided to use sponges as natural samplers because a sponge like this big can actually filter almost an Olympic pool in less than 3 or 4 hours. And that's a lot of water. And I was not super, let's say, positive, because I was thinking that maybe they would process this material really quickly or they would degrade it. But the truth is we use as a proof of concept, we decided to use what I had in the freezer, so samples that were not collected for these purposes in Antarctic waters and also in the Mediterranean. And then we did the regular protocols in an eDNA lab, actually, but the regular protocol of DNA extraction and then amplification of the myfish primers, because we wanted to use something that was very robust. So we focused on the fishes. And then we did just amplicon libraries and metabarcoding processing. And the truth is that they recover a lot of what we knew that were living in these ecosystems, distinguishing them really, really well. So we thought maybe we have something here in our hands, but it was just a proof, and this proof of concept was actually coming from a conversation that we had. We didn't know each other. Stefano Mariani, who is a very well-known researcher in the fish world, but also in the eDNA world, he had an epiphany, I would say, about using sponges as natural samplers, and he contacted me. And after doing this trial with the samples that I had in the freezer, we decided to escalate this to a bigger project, and we got funding for 3 years to actually develop a methodology to use sponges as natural filters. And the reason why we wanted to use sponges as natural samplers is because it's way more replicable than using water, way easier. You don't need special equipment, and there's sponges everywhere in every single ecosystem. I was actually talking to Madeleine before, even in freshwater ecosystems, you have sponges. So everywhere you can find sponges and you can use them as natural samplers. But there was a lot in front of us in terms of optimization and development of the protocols. So the first thing was what is the DNA persistence in the sponges versus the water, because we wanted to implement this as an alternative, sometimes complementary approach to use water, right? So does it persist in the sponges for longer than it does in the water? So we did this in a tank environment in the Horniman Museum in London when I was there working. And in the tanks, we decided to use 3 different sponges that were living happily in these tanks, and they were actually trying to get rid of them. So before getting rid of them, because they were a little bit of a pest, we decided to use them as they were filtering naturally well in these tanks. And then we were putting some fish that we knew which fishes we put in the tanks, and then we were removing the fish, and we took samples of the sponges and the water in these different times, before introducing any fish, after, and then after several hours when we remove the fish. And we sequence both the water and the sponge tissue. And it's true that not all the sponges, you have the results here, and these are the different species of fish that we put in the tanks are in different colors, and then the times that we collected the water and the tissue of the sponges. And here on the left, you have the aquatic eDNA, and on your right, you have the sponge, the eDNA recovered from the tissue of the sponges. And the truth is that both methodologies were relatively similar. Both the water and the sponges recovered the same fishes. I mean, all the fishes that we put in there, but not all the sponges were equally good for recovering the species. Some species of sponges were filtering more and some less. And this is because sponges have lots of microbes inside their bodies, because they are holobionts, and some of them have almost 90% of their bodies completely full of microbes. And these guys get lots of nutrients from these microbes. So these guys don't need to filter that much. And this is what is behind this. But one of the things that we could see is that the DNA in the sponges persisted longer than in the water. So now we know that they are robust natural samplers, and that the DNA persistence is higher in the sponges than in the water. So they're really, really cool. Another thing we wanted to do is to understand, or to test the different methodologies for DNA extraction, if these had any impact on the amount of species that we could recover from the tissue of the sponges. And the truth is that they were all relatively similar, but the best one was this kit that everybody uses in their lab. So it's a very standard way of DNA extraction, that is the QIAGEN blood and tissue kit. So this is a methodology that is replicable, is relatively cheap because collecting sponges is not very expensive, let's say, in coastal areas, and you can use standard lab kits. So we know it's a reliable method, it's easy to implement and relatively cheap, and probably better than using filtering seawater, but we were now ready to escalate this to natural systems, to natural ecosystem. And especially, we were very interested in knowing whether this could be a useful methodology to monitor the deep sea. Because as you may know, sampling in the deep sea is extremely expensive, and sometimes you don't have a lot of time or opportunities to go and collect enough material to do traditional assessment of monitoring biodiversity. So using sponges that are extremely common in the deep sea could be a very effective approach. These are the places in where we know, where we have explored the deep sea. And obviously there is a bias in this map towards the North Atlantic and the coasts or the coast near the United States, right? Obviously, there's been a lot of research in these areas and a bit in the Antarctica, but most of what you see is in black, because that means we don't know anything about what is living there. And people have used traditional surveys that you can see here, where lots of bycatch, you have here that it looks like potatoes or I don't know, even rocks. These are sponges, and these are bycatch that the fishing boats get when they are looking for fish. So all this material can be used for natural sampling of eDNA. Because the other methodology that we have right now that is cost effective is image analysis, but it's really hard because most of what we have in the deep sea, we don't know what it is, and trying to identify these organisms just by a picture is really, really difficult. And there's a lot of development that is very promising around using artificial intelligence to actually understand these identifications, but these artificial intelligence algorithms, they need something to train on, right? And if we don't know anything about what's living in there, they cannot be trained. So our approach could be a very useful tool. And it's, as I was telling you before, it's even more useful, or it could be even more useful, because we know that in the deep sea, there are sponge grounds, and there are also some coral gardens or coral reefs in the deep sea, but there are, I believe, more sponge grounds than anything else in there. And that means that sponges make three-dimensional structures like the coral reefs in the shallow waters that allow many other organisms to live and even to use them as fish nurseries. And these are biodiversity boosters in the areas where sponges live. And sponges also have a very fundamental role in these sponge grounds because they drive biogeochemical cycles. They drive the nitrogen cycle through the symbiosis with their symbionts, also the carbon cycle, and also the silica cycle, because most of the sponges that live in there are siliceous sponges that use a lot of silica in the deep sea. And they also form fantastic habitats. I will show you what a deep sea sponge ground looks like, and there are several examples in this video. Let's see if it plays well. Yeah, you can see here one of these sponge grounds in the Arctic, in the South Massif. And most of what you see are sponges, hexactinellate sponges that are fantastic filters, but you also see many other sea anemones and other organisms living in here. Again, most of what you have on your screen is actually a sponge. So they're very, very, very important for these ecosystems. You can see even fishes in here. These are one of the species that we use that I will show you later. This is a geodia. It looks like a big potato. And sometimes these sponge grounds are monospecific, like this one that you have here that is mostly one species of sponge that dominates, and sometimes they are multi-specific and several other species live in there. So one of the first studies that we did was with Bacella burtalesi, that it's this Russian hat sponge that you have in here that dominates the sponge, or dominates the grounds, the deep sea grounds in the coast of Canada, in front of Halifax, you have here. And there is one singularity of this sponge that that we became aware during another European project that we were doing about sponge grounds. That is, this sponge can settle onto structures that we put in there for recording the noises of fishes in order to do census of these fishes living in there. So these are like structures that we put in the deep sea, and then they are removed because they want to take out the fouling organisms that you have in there. That is mostly sponges. So we were collecting these sponges to use them as natural filters. And the good thing is that we had this census that were happening in these areas, and also there are good estimates from traditional surveys of the biodiversity in these areas. So we took them in several different places across along this gradient where these are placed, and we also took water from the same areas in the surface and also very close to the bottom. And again, we did the similar at the same pipeline from DNA extraction to amplicon with MyFish primers to look for fishes, and sequencing processing, and the regular analysis of community composition. And we found fantastic things. So in orange, you have the different species of fishes that we recovered with through the sponge tissue. And then in dark blue, you have the species recovered in the bottom water. And then in light blue, the ones recovered from the surface water, environmental DNA. And there were more recovered in the sponge tissues than in the bottom, way more than in the bottom water. And even most of what is in the surface water was actually collected by sponges. Not everything, but most of it. And then we looked for these different, we looked for the traditional surveys and the actual fishing, the stock assessments that the Canadian authorities do here. And there were complete matches from what we get in this study. So the visual census, there were 11 species that we couldn't identify, and we had actually 14 that were not identified in the visual census, and only 9 of them were shared between both. And the ones that were in the actual sponge tissue were species that are known that they can actually be in these habitats, but they're not very, very common. So they are like transient. But at this point, I think we were ready for escalating this to understand the complete communities in the deep sea, because it's fine, fishes are fantastic and are very important, but I'm a sponge biologist with an interest in the actual communities living in there, not only the fishes. So we decided to use the same samples of these 3 sponges that we had collected across the different sites that you have in the map, and we treated them through the pipeline with MyFish primers and also with the Lerai primers for CO1. And first, let's focus on the fishes, because they were relatively, they were very, very abundant. And we recovered with the MyFish primers 58 species of fish. And the good thing of this is we recovered these fishes from the areas that we know they inhabit. So there's always a bit of concern about the contamination and stuff that can happen through these pipelines, and it's true, it can happen, but these were very reliable estimates. But as I told you, I was very interested in using CO1 to understand the entire communities. And you have here different pie charts for different species of sponges that we use as samplers. And the best one, because you have the reads that we collect, the reads that we sequence and were assigned to porifera, to sponges, were way more in these different Geodia species than in Fakelia ventilabrum, that we almost recover, that we recover a very small percentage of reads just assigned to the sponges. And this is important because obviously when you sequence the tissue of the sponge, you're going to get a lot of eDNA, a lot of DNA actually from that sponge that you're using as a sampler. And that's very important for the Geodias, but not so important for Facelias. Facelias were super filterers. And we got reads from arthropods, chordates, cnidarians, echinoderms, mollusks, and even sponges, and many other different phyla. And in order to compare the results from the myfish primers and the CO1, we could compare obviously for fish, and we recovered with the CO1 primers 50 species of fish. And as I told you, we recovered them from the areas where we knew they were common and abundant. And we recovered all these different species. And I was very happy to see that they only were present, or they only were detectable in the areas where they are known. But there were obviously some problems. Only 26 species of these 50 something that we recovered with each method were shared between both methods. And this is because of the primary affinities for the different organisms. Some are not easily detectable or easily identifiable with CO1, and some are not with 12S. And this is important because some commercial fish were only detected with 12S. So for fish stock assessment, maybe this is not the best primer set. CO1, I mean, for 12S is fantastic. But if you're interested in the biodiversity of the region, maybe 12S is not the best, because some of the sharks were only detected with the CO1 primers. So maybe the approach, maybe not, but definitely the approach is using several primer sets. But we were interested in more than the fish. And you can see here that living with the sponges, there are lots of cnidarians, extremely beautiful corals or cnidarians that live in these areas. So amazingly, we were able to recover 89 species of cnidarians, of deep sea gorgonians, and even jellyfishes. And again, they were only detected in the areas where they are. So there was not cross-contamination between samples or between libraries, or even from the laboratories where these were done. But we also were very interested in knowing what sponges live in there, because we were collecting sponges from sponge grounds that are full of different sponges. And you can see here that sometimes they are dominated by one single species. This is Fakelia ventilabrum, one single species of sponges, and sometimes there are several more. And we could recover 90 different species of sponges, from deep sea sponges. There are some problems associated to these estimations of biodiversity, and that is that the databases are not fantastic. So sometimes you get species, or you get assignments to species that are actually very well known for tropical areas or places like that, because it's a sister species, like or a relative of that species, but there's nothing else that you can compare your sequence with. So it is extremely promising, but we need to populate the databases with CO1 or with whatever marker we want to use for deep sea organisms. So the take-home message is that sponge eDNA is a fantastic tool, but obviously needs a lot more development. And basically, you need to use several different primers in order to get the whole communities. And if you're interested in fish detection, it's probably best that you use the 12S, or if you have money, with CO1 as well. The next level was trying to understand whether this would be a reliable method to detect invasive species. So we decided to take it to a case study we have in Spain. This is a lagoon, a coastal lagoon that we have that is very, very interesting because it's a lagoon that has sandy beaches around the entire place, but the town and the authorities have placed some wooden docks in them, so people don't have to go through these beaches that are not so sandy, but they're very, I would say, uncomfortable to walk through. So they use these wooden docks to actually swim and be able to get farther away in the sea, so they can go and swim. So these wooden docks are used by many species, sessile species that settle in there, and they're actually stepping stones for invasive species that come with the different boats, because this is it's very close to a very important harbor in Spain. So there's a lot of potential of invasive species in these areas. So we use these sponges that are very beautiful, these pink ones that you have here, several different species of Aliclona that live in these structures. And we did exactly the same. And yes, they are fantastic to identify not only threatened species, because there are many processes that are affecting this coastal lagoon in terms of contamination and in terms of increase of temperatures that are destroying the ecosystems, but also there are lots of invasive species. And we detected many, but I was just going to highlight these two that you have in this picture that was taken in actually one of the close to a sponge that we were sampling. And you can see Botuloides niger, which is an ascidian, and also this polychaete that you have in here, Branchioma bolense. There are two of the most invasive species that we have in these coastal lagoons, and they were detected in almost every sample that we took. So we are now using the same for monitoring the invasive potential that we have in the harbors in Spain and also in some other places in the world, but I don't have results to show you later. And I think that was all I wanted to tell you. Thank you very much.